In chapter 13, we are going to talk about the bacterial genome replication and expression. Now, if we just elaborate the title of the chapter a little bit more, this is basically we are talking about the central dogma of molecular biology, which is basically the information flow in the living system. And this is the same in eukaryotic system as well as the prokaryotic system. The basic system is the same. So the DNA is the genetic material, and it needed to be replicated prior to the entity or the organism divides into two, which we already know. So we are going to learn the bacterial DNA replication, and then after the replication in the genome, in the DNA, all the information for making a functional product is there. And we will mean by the functional product, which could be RNAs and it could be the proteins, which could be structural as well as functional, like enzymes. That is needed to be produced. And in that process, the second part of the central dogma of molecular biology is the information flow from the DNA to make the RNA and maybe the messenger RNA. And to make the messenger RNA from the DNA is the transcription. And this is done by several set of other enzymes. And in the last part of the information flow, that these produced mRNAs are going to get translated by the ribosomes into a functional product of proteins. Now, these proteins could be structural proteins from the organism, these proteins which make up the organism, and these could be the functional protein for the organisms, mainly the enzymes, to carry out all the different reactions. Now, other than this last part, which is making of the proteins by the translation, there are some processes where the information flow can stop at the second level of the transcription. These are making of the transfer RNAs from the transfer RNA genes and making of the ribosomal RNA from the ribosomal RNA genes. These by themselves as the end products. So other than that, what happens that the question is that, okay, all right, in the DNA, we have the information for making these special kind of RNAs as well as for making functional or structural proteins. Now, what about others? For the carbohydrates, other macromolecules that is needed, or the lipids. Now, just remember that once we synthesize the proteins, which are the enzymes, then we can consume all these organic material from outside or synthesize, and the enzymes required for the synthesis or assimilation of carbon is in there. So we can make or we can utilize the carbohydrates, and by we means like the bacteria and all archaea and all eukarya, they can make the other molecules which are carbohydrate or the lipids by different means, because the enzymes required for these processes are supplied by this basic central dogma, last part of the central dogma, which is the translation. So this is how the information flow basically is done. And what we mean by the expression is basically the genetic code that is embedded in the DNA that is basically expressed into a functional product. So this is basically called the expression. That is the end product. That is what we mean by the expression. And that will involve the transcription and the translation. So the first part, we are going to talk about the DNA replication. And this is the outline of the first part. And we are starting with the 13.3. The 13.1 and the 13.2 basically talks about that how DNA was a genetic material that was discovered and the structure of the DNA RNAs. And these are some of the prereqs and basic biology. So we are skipping that. We are starting from the first part of the central dogma, which is of molecular biology, which is the DNA replication in bacteria. So, of course, it's a very complex process and numerous proteins help, and we are not going to that detail of the information that is available out there. We are just going to, still our material will be a little bit challenging. There will be a numerous names and enzymes, But in reality, there are much, much more compared to that. So, first of all, what happens that when these DNAs are replicated, there is a tremendous amount of accuracy in this process. But still, sometimes the enzymes which make a copy of a DNA from a DNA make error. These error rate in bacteria is about 10 to the power minus 9 to 10 to the power minus 10 uh, per base pair. And it is about, say, 10 to the power minus 6 per gene. So per gene in a million times, it will actually mutate. And this actually is pretty good, but it is even much, much more better in the eukaryotes, and particularly for each level of the evolution, the different types of eukaryotics that evolved over the period of time, particularly in the vertebrates and the primates. These accuracy of DNA replication even much, much more robust, make very few mistakes. But still, the mutation is a part of any kind of genetic information replication. So that is there, whatever small it is. This is extremely fast. It's about 1,000 base pair in bacteria is synthesized in a second. And during the replication, the two DNA strands will separate, and each one of them will serve as a template. And the DNA replication is semi-conservative. So here we are looking at a parental DNA strand. They are the two different bases. We know that AT bonds and GC bonds. In between AT, there are two dots, which are denoting two hydrogen bonds. In between GC, this is denoting three hydrogen bonds. So this is a stronger bond. And they are intertwined together into a helix. Now, during the replication, these two strands are separated, and where it's separated, we call this as a replication fork. And as the replication fork is at the site of DNA synthesis, and we're going to see that. And each of these blue strand is the parental strand. And using this parental strand as a template, a complementary new strand is going to be synthesized, which will be the new strand. So now we will get two strands, and each one of them will have a parental strand and a daughter strand. And that is the reason we called DNA replication as the semi-conservative. That means that there is always one strand comes from the parent. So after that, this is a page where I have lined up all the DNA replication proteins. There are more than 30. These are some of the most important ones. That's what in there. So you can just take a look at those. They are all together. And we are going to talk about each one of them. So we are not going to go into the detail of in this page. This is just for your reference, a quick reference, all the proteins in there. So you need to know these different proteins. So first, let's take a look at the overall replication process. So the DNA in the most of the bacteria, they are circular, and a bidirectional replication form from a single origin, which is a 260 base pair region. And here a number of protein actually binds, and the strands actually separate to form the replication fork. And this is where the replication initiation actually happens. And here, this DNA A actually binds a 9 base pair region, and there are 12 times. These are repeated 12 times, and you can see that they are kind of an inverted repeats. These are the direction of the repeats, and these are the direction of repeats, and there are 12 times. Then close to this, there is also another region, which is known as the IHF, which stands for integration host factor. These are also, all these elements actually distort the DNA and basically separate the two strands. And next to IHF there is another region which is called DUE or DU. This is the DNA unwinding element. And this region is 80 base pair rich, and that actually helps for separating the two strands, because as we just said, that in between AT there are just two hydrogen bonds compared to the GC. So it is easier to separate the AT-rich region compared to a GC-rich region. So these initiation sites or the origin sites or the replication sites are usually all the time rich in AT. So this basically causes, as the strands separate in this region, immediately you will see that from each strand there will be a new strand will be synthesized, and there will be a replication fork on each side. There will be a replication fork on each side. So immediately, as the replication fork will separate, the origin will also separate basically. So this is the origin. The origin is in both strands, so it will immediately separate, and these are the replication fork. And this is basically a circle within a circle. So this basically gives what is called a theta structure, like the Greek letter theta. And this is how the replication continues. And what happens that this is one above the other, these two circles. And so we basically have two origin of replication for the two circles, and we will have a single termination site. And this ter site is where the replication will end. And the ter site is the same for the two circles. And finally, what happens that this will lead to the separation of the two circles, and each will have a separate origin of replication and a separate origin or separate termination sites. So this is the overview of the entire process, and we are going to go into the detail of that. So on the other hand, there are some basic idea. The replicon is the portion of the genome that contains an origin and is replicated as a unit. So here, this is a replicon. So after that, what we are looking at the DNA replication initiation, the replisome contains about 12 proteins involved in the replication. And there are two replisomes that moved in either direction away from the origin. And the helicase, and you can see the rings encircle the DNA, and the helicase is responsible for disrupting the hydrogen bonds, which are in between the base pairs, what we call as the AT-rich region, and it will break also the GC region and provides the force to move the replisome, which is just behind this. So after that, the replication machinery, mainly there are polymerases. These polymerases catalyzes the synthesis of complementary strands of the DNA. And when we look at the DNA polymerases, these DNA polymerases are found in prokaryotes, which means that all three domains of life, which are bacteria, archaea, as well as the eukarya. Now, all these DNA polymerases can be classified into different families like A, B, C, D, E, like this. And after this family, they can be again subdivided particularly for the bacteria. We name them as DNA polymerase 1, like a Roman letter 1, DNA polymerase 2, DNA polymerase 3, DNA polymerase 4, and 5. So there are 5 of them, main polymerases in the bacteria. On the eukaryote, on the other hand, we name them with a little bit different way. We actually name them with the Latin words. So we have the DNA polymerase alpha, then beta, gamma, so on. And the most prominent out of them for the replication is usually delta and epsilon. And the DNA alpha is the primase that we're going to talk about. But this is for the eukaryotes. What we are going to see that the DNA polymerase 3 is the main enzyme that will do that. DNA polymerase 1 is responsible for some of the repair. DNA polymerase 2 is responsible for some of the functions as well. We are going to see that what they do. Now, one of the characteristic of the DNA polymerase, and this is true for all of them, the DNA synthesis always occurs in a 5' to 3' direction. And this is the direction of the phosphodiester bond formation. It cannot be formed the other way. So to do this, the template always need to be 3' to 5'. So the template need to be 3' to 5', on which the complementary 5' to 3' is synthesized by the DNA. So it requires a template, a primer, which could be the DNA or RNA strand, and it needs the DNTPs, which is deoxynucleotide triphosphate, which is DATP, DTTP, DCTP, and the DGTP. So basically, you have a deoxyribose sugar, and at the number one carbon of that, there is an adenine, thymine, cytosine, and guanine is added, and then you have three phosphates. In the process of making the phosphodiester bonds, out of these triphosphate, two phosphates actually go out in the reaction, and only one phosphate is incorporated into the DNA chain. So here we are looking at the DNA polymerase reaction. Here you would say that this is your first nucleotide. So right here you have the sugar. This is a pentose sugar. So you have 5 carbon. You have 1, 2, 3, 4, and 5. This is the previous base. And number 1 right here, as I said, is the attached to the base. So here the base is T, or this is your thymine right here. Now you have the 1, 2, and the 3. At the third base pair, you have right here open hydroxyl, which is not shown here, which is added with a phosphate. And here you have the second one, which is also the same, just the base is added in instead of thymine. Now, what you have that we are joining a new nucleotide, which is a cytosine, to this A. So here, this is what is there in the DNA chain or growing DNA chain. The 3' end does have an hydroxyl right here. The incoming nucleotide, which is a cytosine, same structure, the 5' carbon is attached with this triphosphate. You have an alpha phosphate, beta phosphate, and a gamma phosphate. And you can see that this oxygen makes a nucleophilic attack to the alpha phosphate, and then this makes an attack to this oxygen. These two pyrophosphates, or these two basically is this broken down, and the energy is utilized in this process. So these pyrophosphates are released and recycled in the cell. This is now the formation of the bond. You can see that this O basically bond up with this. So this will be basically the next bonding, phosphodiester bonding with A and the C, and the PPI, which is the pyrophosphate. These two are basically released. Now this one does have a 3' end of the chain and one nucleotide longer, but this hydroxyl is open. So any base right here, which could be ATCG, any one of them can come over here and be joined together. So this is what you are seeing. So you can see that the DNA does have a sugar, which is the yellow one, the sugar and the phosphate backbone, and you have the bases which are protruding inside. So as you can see that this is one chain. Basically in the double-stranded DNA, this is the one chain. So this is going from 5' right here and towards the 3' because this 3' is open. So this is your 5' chain. You're going to see that the other complementary strand actually is directed upside down, where the 5' starts from this end and 3' goes in this direction as well. So this is how the two chains are located. So you can see that the two DNA strands are basically anti-parallel to each other. They are anti-parallel to each other. All right, so after that, what we are looking at is the replication machinery, and the E. coli does have 5 DNA polymerases. We talked about that, and the 3 playing the major role. And the DNA polymerase 3 holoenzyme is of 3 core enzyme, and each one of them is basically composed of 3 different proteins and many other different proteins as well. And 2 core enzyme replicate the DNA strand, and here is the 3rd core as well. So here you can see that DNA polymerase 3, this also contains this tau factor, and this tau factor associates the DNA polymerases with the helicase as well as the primase, which basically sets down RNA primers. We're going to see that. There are some, the clamps are associated, which actually helps for loading the DNA into the clamp for processivity, which is also known as a beta clamp. And along with that, which is not shown in here, these 2 DNA polymerase 3s also contains several other proteins, which are alpha, epsilon, and theta, which is not shown. And along with that, what happens that the how actually, again, as I said, connect this to these proteins to even a larger complex. And this larger complex is also known as the clamp loading complex, which acts on these two clamps, which are also known as a beta clamps. And these clamps basically open and close. So you can see that it can open and close. And as it opens, the DNA is channeled inside it, and the clamp loading complex basically allows the DNA to load onto the open clamp, and then this clamp basically closes so the DNA is inside and does not fall off. And the single-stranded DNA is stabilized by the single-strand binding proteins so that they don't, again, base pair or with their complementary strands. Some of the enzymes which are also present in there that we talked about beforehand is not shown in detail is, say, for example, the DNA gyrase. So this DNA gyrase actually relieves the supercoiling of the DNA ahead of the helicase and separates the daughter molecule in the final stages of the replication. So without the DNA gyrase, there will be too much tension on the DNA and it may break. So this is one of the protein which basically keeps as the helicase unwinds it, this actually relieves the supercoiling or the tension in the complex. Now here a little bit of detail of the helicase unwinds the DNA strand, coat the single-stranded DNA to protect the damage, the SSBs, and now you have these topoisomerases. This topoisomerase is actually a class of enzyme, there are topoisomerase 1, 2, 3, 4, and their function is basically to relieve the twist generated by rapid unwinding of the double helix, which prevent supercoiling, and they transiently break and reseals one or both strands without altering the nucleotide sequences. And what we are going to see that the DNA gyrase is actually a class of topoisomerase as well. And then we are also going to see that at the end of the replication, when the two DNA strands are interlocked in between, one circle within the other, the topoisomerase actually also helps to separate these two newly generated DNA molecules. And the primase, that what we are basically seeing right here, this primase synthesize a short complementary strands of RNA primers, approximately 10 nucleotides, needed by the DNA polymerase. And why so? This is the interesting part, that the DNA polymerase cannot start synthesizing DNA replication, or it cannot start DNA replication by its own. And we call this that the DNA replication cannot start DNA replication de novo. De novo synthesis means like from nothing, from scratch. DNA replication cannot, but DNA polymerase cannot do that. So what it needs a push. Say, for example, once your car actually starts, then it can run on the gas, but you need a starter and the battery to start the car first before it, so you need that battery electricity to start the car before it can move on. There's the same thing right here. You need an RNA primer, and the primase can synthesize the primer de novo. That means from the nucleotides that are present in the cytosol, in the cytoplasm, the primase can synthesize a short RNA primer. Primase is nothing but just a form of RNA polymerase, which synthesize about a 10 nucleotide short RNA. So these RNA actually hybridize with the DNA, and this is an RNA-DNA hybrid. And on this RNA, the DNA polymerase takes over and start adding DNA one after another. And at the very end, these RNA primers are removed. This gap is basically repaired and sealed by some other enzymes that we are going to see. So this is the function of the primase, to allow the synthesis of the primer so that the DNA polymerase can start synthesizing the DNA for the DNA replication. So here what we are looking at, that the DNA polymerase can synthesize in 5' to 3' direction only, and that causes a problem. So here you can see that this is the replication fork right here. This is one of the DNA topoisomerase, and you can call this as a DNA gyrase as well, which is behind the helicase. So that your helicase complex is right here. You can see as helicase will unwind the DNA, this will make the tension proper, so that during unwinding of the supercoiling, the DNA doesn't break. So now here in the replication fork, you can easily see that this strand is from 3' to 5', the top one. The bottom strand is, on the other hand, you can see that it is from 5' to 3'. So the top strand, first what happens, which is not shown here, is basically right here, a short RNA primer is first synthesized. Let's take a look at this right here. This is not shown, but right here, a short RNA primer is first synthesized by a primase. And after that, from here, the DNA polymerase takes over and starts synthesizes the strand. And we call this one strand as the leading strand, and the DNA replication is continuous. As the DNA is unwound in the replication fork, this 5' to 3' DNA replication is going on. But the problem that is happening in the other strand, you can see, is 5' to 3'. So if you start replication from this end to this end, this will be 3' to 5', and DNA polymerase does not work in that direction. So what is needed, that the DNA replication on the other strand, which is also known as the lagging strand, the DNA synthesis actually happens from this direction, which we call as a backstitching kind of mechanism. So what happens, that first and small RNA primer is actually put. Now it reached right here, and this RNA primer it was initially right here. After that RNA primer, the DNA polymerase 3, there is usually one here and one here. We are going to see that. And it's going to basically synthesize the DNA. We can just use a different color for that. It can synthesize the DNA, as you can see. And this is known as the lagging strand. After it is a little bit synthesized right here, then what happens? More and more DNA is basically single-stranded DNA is coming in. By that time, this reached the end, more single-stranded DNA is coming in. So there will be another small primer and a DNA synthesis. So then this goes in here. Then will be another small primer and a DNA synthesis. So this is working backwards, as you can easily see. And this actually gives rise to these fragment-like structures, as you can see, which we called as these Okazaki fragments, about 1,000 to 1,200 base pair long, each one of them. And each one of them will have a primer, which is laid down by the primase. Then you have DNA polymerase 3 synthesized strand, DNA polymerase synthesized strand, and there will be a small gap in between them. And as it goes on, there will be again another primer, and so on. So here you can see that on the lagging strand, the DNA synthesis is discontinuous in the form of Okazaki fragment, who discovered this. And after the end, these Okazaki fragments, these primers needed to be removed, these gaps needed to be filled, and they need to seal the gap. So this is basically the continuous and the discontinuous synthesis of the DNA in the leading strand and the lagging strand, and the lagging strand required these Okazaki fragments. So here, this is a little bit bigger picture of the entire part, which is from left to right. You can see that this is a single strand binding protein, and then you have this clamp loader complex. Then you have the leading strand, and this is then you have the primase right here, and then you have the clamp polymerase 3. Then you have the tau helicase. Then here on this side you have the lagging strand, and you have the parental duplex on this. So as this helicase is unwinding this, and your DNA gyrase or the topoisomerase is on this side relieving the tension, on this leading strand, this DNA pol is basically synthesizing after it is the prime is actually set the primer up. This DNA polymerase is synthesizing. See, this is right here. This portion is designated as a primer. Then DNA pol 3 is synthesizing the DNA strand pretty much continuously. On the other hand, what we are going to see that in the lagging strand, the DNA is basically spooled through in a little bit complicated process. So if you carefully look at that, this is the double strand DNA, and this is one strand going in through here. Whereas this is spooled like here all the way up in here. So that's how it is. And you can see that the prime is synthesized this primer, and this will reach here, and see that this is finally this will reach right here. So this is the primer, and the DNA polymerase is now synthesizing this Okazaki fragment. This was the previous Okazaki fragment, and this primer comes in over here, and that is how that each one of them is going to synthesize. So this is a backstitching mechanism, as you can easily see. So right here, primer and synthesize after that. So this is basically the overview of that how each strand is basically synthesized in this process. This is a continuous synthesis, whereas this is a discontinuous synthesis. Discontinuous synthesis. All right. So after that, here is the detail of this process. The DNA A proteins bind to the ORI-C, causing its bending and separation of the strands. We already talked about. DNA B and helicases separate the strand, and the single-stranded binding proteins attach. Primase synthesize RNA primer, lagging leading strand synthesize. The DNA polymerase 1 now remove the primer. So here, this is the 1. Right here. So DNA polymerase 3 is the main enzyme, but DNA polymerase removes the primer and it fills the gap with the DNA. And then the Okazaki fragments and the first primer, which is on the leading strand, just only one is in there. That is also basically removed by the DNA polymerase 1, and the DNA ligase are going to join the two strands. So right here, you can see that this basically shows the parental strands and the lagging strands with the Okazaki fragments. And this will be just Okazaki fragments. You will have many of these kind of structures, whereas in the leading strand you will just only have one. So basically, right here is 3' hydroxyl, and you can see that the DNA polymerase 1 removes the RNA primer, fills the gap, and the nucleotide monophosphate goes up, and the nick remains. So now what happens that right here, so DNA, the only thing that the DNA polymerase 1 is basically doing is filling the gap, but not sealing the gap. And then the DNA ligase comes in, which can use the NAD+, which is nicotinamide diadenine nucleotides. And this is basically, on the other hand, in the process, this is converted into nicotinamide adenine mononucleotide from the dinucleotide. So this basically, there is one adenine that not required for the detail. The DNA ligase at its active site does have a lysine, which needed to be attached with one of the adenine to take part of the reaction. And this adenine is provided by this nicotinamide adenine dinucleotide. While it gives the adenine to the DNA ligase, it is converted into nicotinamide mononucleotide. This reaction can be done by ATP as well. So the adenine can be given from the ATP as well, and the DNA ligase will take that adenine from the ATP to do that reaction. That detail is not necessary. But this is a simple way that it shows the reaction. It's basically, you can see that this is the open hydroxyl end is the same way, and this is basically the gap. This is the next base, and this is basically synthesized. Right here, the gap is already synthesized by the DNA pol 1. This was earlier present as an RNA primer that is basically that is removed by the DNA pol 1, and that gap is filled up by the DNA pol 1 as well. But this joining of the last nucleotide with the DNA did not happen. And this is done by the DNA ligase. Right here, you can see that it creates the phosphodiester bond that join the two fragments. That is basically sealing the gaps. All right. So after that, what we have is the proofreading. And the proofreading is also carried out by DNA polymerase 3. And while the strands are being synthesized, sometimes there is errors. And see that what is happening right here as it is synthesizing right here in this strand, this A, suddenly there is an insertion of an A with a C. And the A and C will not base pair. There will be no hydrogen bonding. So this actually distorts the DNA, and there is a steric disturbance in the DNA. And this will cause the DNA polymerase to stop. And as it stops, the DNA polymerase have a 3' to 5' exonuclease activity. Exo means outside, so at the end, if there is an error, it will be a 3' to 5' exonuclease activity. So the last one can be removed by this exonuclease activity. On the other hand, endonuclease activity can cut the DNA strands in the middle of the strand. But exonuclease can only work at the end. Here it is at the 3' to 5'. That's why it is 3' to 5'. If there is an exonuclease that is working on the 5' to 3', there are exonuclease work in that direction too. So here it is at the 3' end. That's why we call this as a 3' to 5' exonuclease activity of DNA polymerase 3. It removes the nucleotide, which is wrong, and replaces with a G. This is the correct nucleotide, and it moves on. This is efficient, but it is not that big. But still it makes error, and it is like in terms of the number that what we have, it is by 10 to the power minus 9 to minus 10 base pair for bacteria, which is very less, but still when you just see the trillions of bacteria that can be made within a short period of time, you can see the number of mutations that can accumulate. It is just all basically those comes back to those number. And finally, what it shows is the termination of the replication. And replication stops when the replisome basically joins, reaches the termination or the terse site. And this forms what is called these catenanes. And these are double-stranded, these are catenated chromosomes, which are a ring within a ring. So these must be separated out. And there is a particular word that we use with this. This is known as terminal resolution. And remember the term, this is known as a terminal resolution. And the term terminal resolution in DNA replication means that how the the two newly synthesized DNA molecules, how they are separated. That is known as the terminal resolution. And the topoisomerases basically break. This is a double-stranded break and rejoin the DNA molecules. And this is the overview of that. And one of the things that there are also some enzymes which are called the recombinase enzymes, which XRCD is one of them. And this catalyzes an intramolecular crossover that basically separate the two chromosomes. So here you can easily see the blue chromosome is basically intact in this, and the red one is nicked and separated out. So that's basically, on the other hand, the process of the termination of replication in the E. coli. And so this is by the topoisomerase, and this is by the XRCD, where you can see that there are two dimerized chromosomes right here, and right here, the crossing over occurs at the termination site, and the resolution of the dimerized chromosomes are basically achieved by the XRCD. This is by the catenanes, by the two types. The catenanes are basically separated by that topoisomerases, where dimerized chromosomes are separated by the XRCD recombinase enzymes. So after that, we are looking at that the bacterial genes and what are the different types of genes and what are the different coding regions and important sequences. So the gene encode the different product, and historically there were lots of different definition for genes. And at one point of time, we used to know in molecular biology that one gene, one protein hypothesis. But one gene, one protein is also not the correct word, because from one gene, we can make the transfer RNAs, mRNAs, and the ribosomal RNAs. And the ribosomal RNAs and the tRNAs are the end product to do some function. On the other hand, that mRNA goes onto the translation to form the protein as well. So protein as well as RNA, both are products. And now we have modified that definition of the gene. We can either say that genes encode proteins and RNAs, or you can say that it produces a functional product, basically, because it is basically forms the mRNA, which is then translated by the ribosome to make a protein. So let's take a look at that. What are the different regions of a gene? And the first, what we have is there are two strands that you are seeing. This is the 5' to 3' strand. This is the 3' to 5' strand. The 5' to 3' strand is known as a sense strand. Also, this is known as a coding strand. The why we call this as a coding strand as well, because the nucleotide triplet that you are going to see in here, that will be almost the same three nucleotides that you will see in the mRNA that is produced from here, that only the T's will be replaced by the U's. On the other hand, the other strand is known as the complementary strand, and this complementary strand is also known as the antisense strand. And this complementary strand is basically the template strand, because when RNA is synthesized, the RNA polymerase, which synthesizes the RNA, that's going to read this template strand from 3' to 5' direction and synthesize the product in 5' to 3' direction. So just as the DNA polymerase can only synthesize from 5' to 3' direction, RNA polymerase can synthesize also in 5' to 3' direction. So it has to use this template strand to synthesize the RNA. The 5' to 3' strand just separates out in that process, and it doesn't have the function directly, because the RNA that is produced is just single strand. So there is no lagging strand or Kazaki fragments or anything like that, because this only one RNA strand is going to be synthesized using the template DNA strand. So what are the different regions of a gene? So you can see that the gene actually starts from this +1 site right here. This is the start of the gene. But upstream of the gene, which is right here, before the start site, there is some region which is called this -10 and the -35 region. And these are some of the very important sites where the RNA polymerase actually come in and bind. These are some of the promoter regions, which is very, very important. And we're going to talk about this. So these are the promoter regions. And from the +1 region, that is not the start, if it is a protein, of making the functional protein. You can see that right here, if it is a protein product is formed from this gene, this ATG is the first start codon for making a protein. So the protein sequence will start from this nucleotide sequences right here. So from the +1 to ATG, this region in untranslated, and we call this as a leader. This is the leader. And then this is the start, and this is the coding region. You can easily see that this one is a coding region. And then there is a stop, basically. So here the coding region will stop. And then also after that, there is a trailer and a terminator sequence. So you can easily see this is the direction of the transcription. So in the process of the transcription, the RNA polymerase will move in this way, moving out the mRNA, and this is right here. So here you can see that right here, this is the leader, which can start from with a G or A right here. This is the leader. This is a specific sequence which is known as a Shine-Dalgarno sequence, which binds to the ribosomes. And then you have a start codon, which is the AUG. This is the translation start site. And after that right here is a translation stop codon, followed by a trailer and the 3' end. And many times, so this is basically the mRNA that is produced from the protein coding genes as such. Why we are saying that this is a protein coding gene? Because we have a start and the stop codon, which is leading to a coding region of the protein. And we are going to talk about the RNA coding gene soon, pretty much. So basically what happens that the same nucleotide sequence as the mRNA or tRNA, ribosomal RNA, except in basically the DNA basis. So right here, these details are in there, what we already talked about, located at the start of the gene, recognition binding of the RNA polymerase, and orients the polymerase. And leader sequence transcribed and not translated. Shine-Dalgarno sequence important for translation initiation, as we talked about that. And the coding region begins with the DNA sequence 3' 5 TAC 5' produces the codon AUG, and ends with a stop codon immediately followed by the trailer, which prepares the RNA polymerase for the terminator sequence, where it falls off the DNA and releasing the mRNA. So on the other hand, the actively growing cells also need a ready supply of the transfer RNAs, which are basically the vehicles to take the all different amino acids. There are 20 of them at the site of protein synthesis, which is the ribosome. And the ribosomal RNAs are basically the structural proteins of the ribosome. And there are more than one gene codes for a single type of tRNA. And these genes coding tRNA are transcribed from a single promoter. And this controls the amount of tRNA, ribosomal RNA in the cell. So the molar concentration is controlled by this process. And when more of one tRNA is transcribed from a promoter, they are separated by short spacer sequences as well, which we are going to see right here. So here the top illustration actually shows basically these two clover leaf-like structure, and there are two tRNAs separated by a spacer. And one of the arm is right here at the extreme end is labeled as anticodon. And right here is another anticodon. And depending upon what kind of anticodon you have, right here is the amino acid arm, which basically will carry an amino acid, will be specific. Depending upon the anticodon, a specific amino acid will be attached here. Depending upon the anticodon, a specific amino acid will be attached here as well. And here you can see that this is a tRNA. If you have an anticodon AGU, this will put a serine right here. If you have an anticodon GGU, this will put a threonine in here. So they are all correspond to that. So now you can see that this anticodon can read the codon, and if the codon has to be specific for threonine, the codon has to be specific for the serine, and that's how the tRNA will deliver the proper amino acid as per the codon on the mRNA. And here also what you can see that the ribosomal RNAs, which are the 16S ribosomal RNAs, 23S ribosomal RNA, or 5S, they all have a very extensive secondary structure. They can be present on the, usually they have more than one rRNA gene, and they also do have promoter, trailer, and the terminator. And on the other hand, what happens, sometimes this trailer and the spacer of these ribosomal gene can code for the tRNA. So here you can see that in between the 16 and the 23S RNA coding region of the gene, there is a spacer tRNA. On the other hand, this is after this 23S and the 5S. These are all part of the ribosome, small, large subunits. You can see there is another trailer tRNA genes. So they can come in all different types of modification. And These post-transcriptional modification are usually used by the ribonucleases or the ribozymes. So once these structures are basically formed, there are ribozymes, which are the catalytic RNA. They can separate these different 16S, 23S, 5S spacer tRNAs. They can cut and chop these separate out so that they are all separated out and go there on their way to do the different function. And ribonucleases basically does that. So next we are looking at the transcriptional process of the bacteria. So RNA synthesis from the DNA template, we already talked about that. RNA produce a complementary sequence to the template DNA, and three types of RNAs are produced. tRNA carries the amino acid during protein synthesis. Ribosomal RNA molecules are component of the ribosome, and mRNA carries the message for protein synthesis. So what is an operon? Now, the bacterial genes encoding the proteins involved in related processes are transcribed by a single promoter. And this is not common in eukaryotes. The eukaryotes do have a monocistronic mRNA. That means you have one promoter, one gene. Whereas what you are looking at, that the on the particularly for the bacteria, you may have polycistronic mRNAs, which does have a single promoter, and you have coding region. So here you can see that this is a leader region, and you have a coding region 1, you have a spacer coding region 2. These two genes are basically related. So that, say for an example, sometimes we are going to see that in the next chapter as well, that suppose there is a gene which is needed for lactose uptake. So one, it can code for a lactose transporter on the plasma membrane. Another can code for the metabolically convert the lactose into some different form, like an allolactose, and related to different things. So they are related genes. They can be on the same polycistronic mRNAs. On the other hand, the monocistronic mRNAs, which we basically see is in the eukaryote, you have one gene particularly under one of the one promoter, so it basically produces just one functional product from here. So the operon is basically where you have multiple genes or polycistronic mRNAs, and we call this process an operon because there is this one operation going on by a number of genes, basically. And we are going to basically talk about this entirely in the next chapter. So let's talk about the bacterial RNA polymerase. And there are also many types of bacterial RNA polymerases, and they contain the 5 different proteins. And the core is composed of 5 polypeptides. You have 2 alpha subunits, 2 beta, and one is called a beta, and another called is a beta prime. And you have the omega. So 2 alpha subunits, then beta prime, and omega 5 altogether. And also there is a sigma factor attached to it, and this catalyzes the RNA synthesis. And the sigma factor by itself doesn't have any catalytic activity, but helps the core enzyme recognize the start of the gene. So the RNA polymerase holoenzyme is this core enzyme, which is 2 alpha, beta, beta prime, and omega. And the sigma factor makes the RNA polymerase holoenzyme. And only in the holoenzyme in the presence of the sigma factor, it can synthesize the RNA, or it can begin transcription. So here is the process of the transcription. Here we are looking at the RNA polymerase core enzyme. Sigma factor comes in, and it now becomes the holoenzyme competent for the DNA replication. It basically see the promoter region. So basically it binds and scans the DNA, and it will lodge itself where there is a promoter. And the sigma factor actually helps direct the RNA polymerase to the -35 promoter sequence. And once it is directed there, the RNA polymerase actually denatures a short stretch of the DNA at the -10 region, which is AT rich, forming an open complex. And that is basically stabilized by that sigma region in this process. So you can see that the 2 strands are separated. And then, now what happens that now the RNA polymerase core enzyme will synthesize the RNA, and this is shown in red. And once about 12 ribonucleotides or RNA is RNA nucleotides or ribonucleotides are synthesized, and they are all linked. The transcription enters from the initiation phase to the elongation phase, and the sigma factor is actually released in that process, and the synthesis of the mRNA can go on, and you can see that it can go on until the RNA polymerase reach what is the terminator sequence, and the polymerase sees the transcription, and the RNA is basically released. So that is your initiation, elongation, and termination, 3 phases of the transcription. So here, you can see the sigma factor helped to position the core enzyme in the promoter, which is this TTGCA, that kind of region. This is in the -35 base pair sequences upstream from the gene. Then you have a little gap in here, and the RNA polymerase first bind in here and settle here. And then what you can see that this is a -10 sequences, and this is basically the TATAAAT, and this is known as a Tata box in eukaryotes, and sometimes this is known as the Pribnow box. This is an older term called Pribnow, PRIBNOW, Pribnow box in the bacteria, which is basically an AT-rich sequence. Why AT-rich? Because it is easy to melt this part, because you have only 2 hydrogen bonds instead of 3 hydrogen bonds in between the GC. So this is where the DNA start to separate, and which we called as a transcription bubble. So at this point, the RNA polymerase will basically start reading the 3' to 5' template strand and start synthesizing the 5' to 3' RNA by the process of transcription. So here you are seeing that -35, -10 region highlighted, binding of the RNA polymerase hollow enzyme. Right here is the sigma factor. This is the RNA polymerase core enzyme that is forming an open complex, which is the transcription bubble. And you can see the RNA transcript is formed. Once this transcript reaches about 12 base pairs, ribonucleotides, the sigma factor is dissociates, and the elongation phase continues in the direction of transcription. This is basically the initiation part. So the elongation part is basically the extension of that. After binding, the transcription bubble basically moves the RNA polymerase as it synthesizes the mRNA, and within the bubble, a temporary RNA-DNA hybrid is basically formed. And you can see that all the ribonucleotide triphosphates, which are the ATP, GTP, CTP, and the UTP, are incorporated into the RNA, complementary to the DNA template. So here is just a small thing, just always remember that your ribonucleotide triphosphates are different from the deoxyribonucleotide triphosphate, which is used in the DNA synthesis. The only difference is that, remember that, the sugar that is present in the ATP, GTP, CTP, and the UTP is a ribose sugar, whereas in the DNA, what is incorporated that contains a deoxyribose sugar. And this detail is not necessary, but if you just remember the sugar that is present that we basically drew in 2 cases, there is a very subtle difference with those. At the 2' hydroxyl region, there is an OH right here. So this is right here is a C, that is right here C, and you can see that this is 1, 2, 3, 4, and 5 carbon of the 5-carbon pentose sugar. So here, if you just have an OH in the ribose sugar, this is your ribose sugar, and this is basically in the RNA. This is present in these. On the other hand, instead of having a hydroxyl in the DNA, which is the deoxyribose sugar, what you have is just a hydrogen at the second base. So this O is gone from here, and that's why we call it as a deoxyribose. So these are two differences in between. This is a ribose sugar with a hydroxyl at the second carbon. This is just with only a hydrogen in a deoxyribose. And these subtle differences allow the RNA polymerase only to pick up the RTPs or the RNA nucleotides, ATP, GTP, CTP, and the UTP. And this only subtle difference allows the DNA polymerase to pick up the deoxyribonucleotides, which is, instead of this, will be a DATP, DGTP, DCTP, and DTTP. There will be no uracil in there. So there is a small d in front of all of them due to the deoxyribose sugar. So this is just one of the details of that you can just keep in mind. So here the transcription elongation goes on 5' to 3' direction, as you can see, as this is fed through here, and as the direction of transcription goes on, this will produce the RNA, and it will basically reach near the termination site. Now the termination occurs when the core RNA dissociates from the DNA template. And there are two types. One termination mechanism, what we called as the intrinsic termination, or which you called as a factor independent transmission. In this process, what you have that there is a region which is an A-rich region, and it forms a stem loop structure. And that is followed by some region which is rich in this UA, lots of U residues, multiple U residues. So what happens that when the RNA polymerase actually reaches the stem loop, this forms the RNA polymerase basically stem loops destabilizes these UA bonds, and when the RNA polymerase basically reach here, it actually tends to fall off from this and release the RNA polymerase and the mRNA and the DNA. So this is basically the factor independent termination or intrinsic termination by the help of a stem loop followed by U sequences. So sometimes there is also not required. There is a protein factor involved in there, which is called NUS, N-U-S, NUS protein. That is also is associated with this U-rich region, which also helps in the intrinsic termination of this process. On the other hand, the next one is the factor dependent termination, and this is actually observed in many cases. So what happens that there is a site where a factor actually binds and helps the transcription termination. So this factor is known as a Rho, and it can bind to a particular site which is known as a RUT or Rho utilization site. So what happens that RNA polymerase actually is moving along the DNA, making the replication bubble and making the mRNA, and this is the RUT site which recruits the Rho, and it moves and from here the Rho factor actually follows over the RNA behind the RNA polymerase, and the Rho uses the energy supplied by the ATP, move along the mRNA, and it tries to catch up the RNA polymerase, but this is slower than the RNA polymerase, and it cannot catch up easily. Only if the RNA polymerase actually pause, which is a Rho-dependent pause site, only at that time that this Rho factor can basically catch up. And when this actually happens, which is when the RNA polymerase basically pauses, the Rho actually catches up with the RNA polymerase, and that actually associates the RNA polymerase to dissociate from the DNA. And this is basically the specific sequence on the DNA, which is basically the Rho-dependent pause site for the RNA polymerase. So this is Rho factor dependent transcription termination. So after that, we will review the genetic code that consists of the 3-letter words. So here, what we are looking at, the final step is the expression of the protein encoding gene, and we need to know this. the mRNA is translated in the amino acid sequence of the polypeptide chain, and how this basically happened. So how all the information is in the DNA and how it is being translated. So in the DNA strand, which is the sense strand, also we call this a coding strand, and you can see that you have an ACT, GCC, and all along. The template strand, on the other hand, has a TGA, CGG, and so on. So when the transcription is happening, the template strand is read and the mRNA is produced. So how it is happening right here? So look at the 3' to 5' strand and make a complement of that. So if it is a T, it is complement A. If it's a G, it is C, A, U, C, G, G, C, G, C, and so on. There is also another very interesting way that this is, you made the complementary strand of the template strand to make the mRNA. This is the correct way, and this is how the RNA polymerase do it. This is how the polymerase read it and do it. But there is also another way that you can also think about this way to shortcut this process. The coding strand or the sense strand will have the exact sequence of the mRNA, only the U's will be replaced by the T. And see that. It's A, C, C. Here it is T. Instead of T, it is U. Then you have a G, G, C, 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 A. Then you have a T instead of U. So that's why we call this a coding strand, because the sequence in the coding strand exactly matches the sequence of the mRNA, only the T's are replaced by the U's. And now what happens that these sequences, or these 3-letter words that we have on the DNA, they are usually called triplets. On the other hand, those three-letter words, basically the triplets, when they are basically becoming the 3-letter word in the mRNA, we call them codons. So the codons are only in the RNA. In the DNA, they are triplets. So what happens that each codon can be recognized by a specific tRNA, which will have an anticodon sequence, which is basically, think about a decoder. This is the coder, this is the decoder, and that decoder only knows that if you have this one, I am giving you this amino acid. If you have this code, I am giving you this amino acid because I am decoding it with this. You have a coding sequence here, I have a decoder, I'm giving you this amino acid. So the first one, if you have an AUG, the corresponding tRNA which can bind to the AUG will have a complementary anticodon, which is A, that's why it's U, then U, A, G, C. If you have a tRNA where the anticodon arm that we already saw beforehand contains UAC, it must have a methionine attached to its amino acid arm. Right here, the next one, if you have an AGC, you must have an AUGC CG anticodon. And if you have an anticodon UCG, you must have a serine associated with the amino acid arm of that tRNA. So depending upon the anticodons, every amino acid is basically fixed. And that's why you can see from the DNA to the mRNA to the translation, exact sequence of these proteins are embedded in your genetic code. And this is basically the expression of the genetic code through the transcription and the translation. What code is in there? An ultimate expression in the form of protein, which is a specific sequence of the amino acid making a polypeptide or a protein, which will give rise to these thousands and thousands of proteins in eukaryotes and prokaryotes, or the bacteria and archaea as well. So next, what we are looking at, right here, some word right here, 3 nucleotides, specific single amino acid. Reading frame is how it is read. So just remember that reading frame is very important, because if you just do not read the sentence in a proper way, and if you shift it one, then it will not read it. It will cause mutation, and we're going to see that. The start site is always AUG, and the stop, which are called the nonsense codons, these are 3 of them, UGA, UAG, and UAA. They do not encode any amino acids. So there is no tRNA corresponding to these, or there is no anticodon against these codons. So there are basically 64 possible codons, and the 61 codons specify amino acids, including AUG, and these three of them are the nonsense or the stop codons. So that's basically. So on the other hand, up to 6 different codons can code a single amino acid. So here what we are looking at the genetic code, the table. So here you can see this is the first base of the codon, this is the second, and this is the third. So here if you have an UUU, it tells you to bring a phenylalanine. If you have an UUC, it's still phenylalanine. On the other hand, if you just see that right here, leucine, if you have CUU, CUC, CUA, CUG, all these 4 codons code for leucine. So on the other hand, you can see that methionine, MAT, only AUG codes for methionine. On the other hand, here you can see right here, this tryptophan, only UCG code for the tryptophan. But on the other hand, say arginine, there are 2 codons, glycine, there are 4 codons, aspartic acid, there are 2 codons, alanine, there are 4 codons, threonine, there are 4, proline, there are 4, serine, there are 4. So what's going on? Some of them are making 2, some of them 1, some of them 4. So the reason is that if you see that a codon is basically have 4 different codons for a single amino acid, like valine, serine, proline, threonine, alanine, that means that these are the amino acids which are most common in any kind of protein, valine, alanine, threonine. And then the ones with 2, like phenylalanine, leucine, they are the next most common in there with 2 codons. And the ones which are, say, arginine also has 4 codons in here. And serine, if you just look at that, serine, this is right here, 1, 2, and then 1, 2, 3, 4, 5. So 1, 2, 3, 4, and then you have 5, 6. So 6 codons actually code for serine. So which is the most common out of all? There is no other amino acid which is coded by 6 codons. This is the highest. Rest highest is the serine. On the other hand, what happens is that the ones with the 2s, they are a little bit less common, and with the 1, which is least common. And the AUG is methionine that is very specific. Methionine can be in the middle of a protein as well, but as it is always the start codon, there is only 1, so that there is no mistake. And tryptophan is, on the other hand, is very rare, so that's why it is basically coded by just only one codon. So what happens that this actually gives you something which we called as a code degeneracy. So up to 6 different codons can code for a single amino acid, and that is serine. So what happens that any of these amino acid can code for this, and sometimes due to that if there is a mutation that is going on in somewhere, and sometimes that is tolerated to some extent, and this process is basically known as the code degeneracy, which is basically not 6 different, that is specific case, but code degeneracy by definition is that multiple codon can code for the same amino acid, is known for code degeneracy of the codons. So after that is the loose base pairing, which is known as a wobble. And it's important is that when you look at the codons, the first 2 codons are actually important compared to the third one. And this eliminates the need for a unique tRNA for each codon. And this decreases the effect of submutation. So here you can see you have a GGU and you have a C and C that brings in a glycine. On the other hand, you can see that this is a GGC. This is a proper alignment right here. This is wrong. This also brings in a glycine. So both can fit in there to some extent if the first 2 fits. And how this is actually tolerated? Carefully look at that. If I'm just taking the example of, say, isoleucine. So right here, you can see that this is AU. The first 2 are the same. The only difference is in the third one. Say on the other hand, if I just look at the serine, see that all of them are UC. The only difference is the third one, UCAG. So the first base pair, if they basically fit, they can put the correct one as well. So sometimes they can put wrong amino acid, of course, the possibility in there. But again, what happens that the problem, this is the most important problem, that what happens, the mutation that can happen at the level of the DNA, that is the most dangerous part, because that is going to be permanent, because everything the mRNA is going to be made will have that mistake. On the other hand, if you have a mistake while making the mRNA, or even while making the protein like this, if there is a mistake, that is not a detrimental mistake, because if that mRNA make a sequence which will not put a proper amino acid, or in the translation step, if a wrong amino acid is put, and if it makes the protein non-functional, that protein is going to be degenerated, or that mRNA is not going to be no good, that's going to get degenerated. That's not going to affect the life or the well-being of that organism. But on the other hand, if there is a change in the DNA, then every mRNA will be wrong. Every translation will be wrong. But at the mRNA, if it is a little bit wrong, only that mRNA is wrong. That doesn't mean that every mRNA will be wrong by the RNA polymerases. So that's one of the important thing to remember, that why the mutation happens always in the DNA, and we never talk about the problems in the RNA transcription or the translation, because if there are a couple of things wrong, they are just going to be degenerated, doesn't affect the health of the organism. There are some exceptions to these codes. So here you can see right here, there are some selenocysteine and pyrolysines. Mycoplasma uses the stop codon to code for glutamine, and they incorporate two rare amino acids in the polypeptides by using these type of rare amino acid in the protein as well. So the next part is basically the translation of the bacteria. This is the last part of the information flow. So we just talked about that, how the tRNAs can recognize the codons, bring in the amino acid compared to that, and how they are translated into the protein. So here we are looking at the structure of the ribosome. You have a 30S subunit and a 50S subunit. 30S contain a 16S ribosomal RNA, 21 polypeptide, and it's 0.9 into 10 to the power 6 Dalton. So 0.9 megadalton in molecular weight. 50S contains a 5S ribosomal RNA, 23S ribosomal RNA, and 34 polypeptide chains, and 1.8 into 10 to the power 8 Daltons. So altogether, you can see that the 70S ribosome, 30S plus 50S, not additive, remember that. This is your bacterial ribosome, which is different from the eukaryotic ribosome, is 2.8 into 10 to the power 6 Dalton. So 2.8 megadalton is the molecular weight of the entire structure. So here you can see right here, the ribosome is basically the translation, and the direction is N to C terminal. So here we are looking at the tRNA structure. Here you can see that this is called a D arm. This is the anticodon arm with an anticodon loop, which will recognize the codon. And right here you have a variable arm, and then we have a T-Cy C arm. And why we call it T-Cy C arm? Because there are some, you can see that there are codons, well, not codons, there are some bases which are written as a C, and these are the pseudo-uridine. These are a little bit different than the regular uridine. There are some modification, and these pseudo-uridines, and that's why we call this as a T-Cy C loop. They all have different functions in there and beyond the scope of this course. And then is right here is the acceptor arm, and at the end of the acceptor arm, the specific amino acid is attached right here. So that's basically the other. So this is your schematic, and like a tRNA in the secondary structure, that how it shows, which is about 70 to 95 nucleotide long, and We already talked about that. Base pairing within the tRNA molecule forms a cloverleaf structure, which folds into an L-shaped structure, and it's complementary to mRNA codon located on the anticodon arm. So here is how the amino acid activation happens or attachment happens. Enzyme, which is required, very important, is known as amino acid tRNA synthetases, and this is happening in the cell all the time. So what you are seeing right here is the acceptor arm, and you can see right here is a terminal phosphate and an adenine right here. And this will be attached with a high energy bond linking this amino acid to the tRNA. And the amino acid tRNA synthesis need the ATP hydrolysis to do this. And this is basically the carrying arm. Now what you are going to see that this bond will be broken and the amino acid will be attached to the growing polypeptide chain, and this tRNA will be empty. This will go back into the cytosol from the ribosome, and the next amino acid tRNA synthesis will attach the next amino acid to that, which will be the same type. If you have the same codon, each time the amino acid synthetase, suppose this is glutamine, each time a glutamine will be loaded by the amino acid tRNA synthetase on this. So when amino acid is loaded onto a tRNA, we called it the amino acid is charged. So it's a charged amino acid. And when it is unloaded, we call that this is an empty or discharged amino acid, which will again go back. So we have at least 20 amino acid tRNA synthetases, each specific for a single amino acid. And the tRNA is attached to a specific amino acid, or which you call as a cognate tRNAs. So now the ribosome contains three different sites. One we called as an acceptor site. Sometimes we also call that amino acid site because it just brings the amino acid tRNA, and this receives the tRNA carrying the amino acid. And the acceptor site, as is an amino acid tRNA, which means like this is done by the amino acid tRNA. The acceptor site is sometimes called that amino acid site as well, or acceptor site. This is accepting the tRNA carrying the amino acid. The next side of that is called the peptidyl site. This holds the tRNA attached to the growing polypeptide. And then you have the exit site, which is basically the empty tRNA, which is discharged, as I said, goes to this side, and it leaves the ribosome to be charged again by the amino acid tRNA synthetase to bring more amino acids. So here, what happens? 60S ribosomal RNA is needed for the initiation of the translation, and it binds to this Shine-Dalgarno sequence, which is the ribosome binding site that we talked about before. And this also binds to the 3' of the CCA end of the amino acid tRNA. And the 23S ribosomal RNA is a ribozyme or a catalytic RNA that catalyzes the peptide bond formation, which one of the most important process. So the first part, what happens is the initiation of the protein synthesis, and involves the ribosome subunit and other molecules, and it forms an N-formyl methynyl tRNA. This is the bacterial initiator tRNA. On the other hand, the archaea eukarya use just methionine tRNA. So this is so the bacterial initiator tRNA is known as an F-met formyl methionine mRNA. It's just a little bit modified methionine instead of just a methionine. And then you need the 30S initiation complex tRNA mRNA, 30S ribosomal unit, and you need the IF1 and IF2 initiation factor, and the GTP catalysis is required for this process. So what is happening right here? That right here is the 30S subunit in a dissociated stage, which is bound with an IF3, initiation factor 3. This prevents the 30S subunit to bind to the 50S subunit. Now, in the initiation, there is something going on here. The initiator tRNA is the F-met, and what happens that a GTP is bound to this IF2, and this IF2 GTP F-met tRNA makes what we call as a ternary complex. You have a IF2, GTP, and FMET tRNA. Three things, this ternary complex. These ternary complex is recruited onto the 30th site, and the mRNA also comes in and binds with the FMET tRNA because it contains the AUG right here, and it stays in here in this process. So as soon as this binding actually occurs, the IF3, which is the preventer or inhibitor of binding the 50S 50S ribosomal subunit, that leaves. So now what you have, there is also another initiation factor that comes in, which is the IF1. So IF1 binds to the 30S initiation complex. So this ternary complex, mRNA and IF1, this is the 30S initiation complex. And now the 50S subunit can bind to the 30S subunit. And this is the GTP. Now what happens? That the GTP bound to the IF2, that is hydrolyzed, and the IF1, IF2 is also released. And here you can see that right here, this is IF2 with the GTP and bound hydrolysis, that this will be hydrolyzed to GDP. So now you have the 70S initiation complex, and you can see that at the P site, which is the peptidyl site that we just talked about right here, this is where the first tRNA is sitting with the formylmethionine in FMET, the first initiator or the first amino acid is sitting in here. The E site, exit site, or the acceptor amino acid site is still empty. So this is the initiation. So after that, elongation is a sequential addition of the amino acids to the growing polypeptide, consists of three phases: amino acid tRNA binding, transpeptidation reaction, and translocation, and involves several elongation factors, which are known as the EFs. Just remember, these are IFs, initiation factor. These are EFs, or elongation factors. So in this process, what happens, that there is a factor which is known as EFTU, which is bound to the GTP, and it binds to the amino acid tRNA, whatever it is, and this will bring one of the tRNA. So basically what happens, that there is a codon right here, and lots of tRNAs are coming in and scanning this site constantly, and only one will properly bind. So once the proper binding of the anticodon recognizes the codon, the anticodon of the tRNA recognizes the codon, the EFTU is released, and the GTP is hydrolyzed to the GTP. And the GDP bound GDP is separated out by the EFTs, and the EFT is then the EFT basically separate from the EFTU, which can again be charged with a GTP, and this cycle basically goes on. So the EFTU GDP hydrolysis to EFTU, this is basically the process of bringing in the amino acid tRNA to the acceptor site. So now what you have amino acid which is bound to the second one. And in the first position, which is the peptidyl, the site right here, the P site, you have an amino acid. So now what happened is a peptide bond formation. So the peptidyl site, this amino acids which are attached to this tRNA is transferred to the third one, and this is known as the transpeptidation reaction. So right here, this is now the three. So here there were two. There were two amino acids. They are transferred. They are separated from the tRNA and transferred to this. So you have now 1, 2, and 3 sequentially. And this is catalyzed by the 23S ribosomal RNA. So now what happens, that you have the this at the P side, this tRNA is now empty because transpeptidation reaction happens. Now that there is another, which is an EFG, elongation factor G GTP complex comes in, and this GTP gives the power to basically translocate this whole structure in a ratchet-like motion from here towards this side. So ribosome moves basically this way, so that this is ratcheted through the ribosome on the left-hand side. So right here, you can see from the P side, this empty tRNA reached the exit site. From the amino acid or acceptor site, this bound three amino acid with the tRNA. Now they are in the peptidyl P site, and the A site, which was occupied by this, is now empty. So what happens? This is powered by the GTP hydrolysis. So this is, and then what happens that as this tRNA enters the exit site, you can see that the tRNA is released from the exit site. This is the discharged tRNA that will go back again to be charged by the amino acid tRNA synthetases and can bring another amino acid. So this is the elongation cycle. So we already talked about that, catalyzed by the peptidyl transferases in this process, and no extra energy is basically required in this process. And the bond linking the amino acid to tRNA releases the enough energy in this process. So here you can see that how this is happening. And if you just carefully look at that, what we are looking at. So we are looking, this is we are looking at the reaction upside down. Here, see that tRNA is at the bottom and the amino acids are on the top. But when we are looking right here, this is basically the tRNA, where you can see that this is right here is the ribose sugar and the phosphate, and that this is the tRNA. And right here is the amino acid. So here you can see your amino acid 1, amino acid 2. And here is the A site, which is the amino acid going to be the amino acid 3. So here you can see this NH2, this amine bond basically attacks this carbon, and then it just breaks this bond right here by a nucleophilic attack. And now this is empty, and the bond is formed in between this. So this is a CN bond that is basically formed right here. This is the transpeptidation reaction, and this CN bond formation is done by the 23S ribosomal RNA. And now this is the part of the tRNA which is empty that would go into the exit site and go on. This is now the tRNA. This is the top portion is the tRNA. And now you have 1, 2, and 3 amino acids here. So this is right here, NHCHR1. This is N, then N plus 1, then N plus 2. These are the 3 amino acids. Now this tRNA attached to the 3 amino acids are going to move into the P site. This one is going to move into the exit sites and go out. So once this go into the P site, there will be a new one which will come into the A site, and this will again happen, and you will have an N plus 3, N plus 4, N plus 4, and that's how the chain will basically grow. So the final phase is a 3 simultaneous event. Peptidyl tRNA moves from A to P site. Ribosome moves down 1 codon, so the new codon is positioned to the A site. We talked about that. Empty tRNA moving from the E site, and this requires the power from the GTP hydrolysis. And then the final one is the termination of the protein synthesis. Takes place this when the nonsense codons or stop codons, UAA, they are reached, and there are releasing factors aid in the recognition of the stop codons. And there is no cognate tRNAs for the stop codon, so the ribosome basically halts. And the peptidyl transferase hydrolyze the bond linking the polypeptide and the tRNA, and the polypeptide and the tRNA are basically released. Now, the selenocysteine and the pyrrolysine, these are the 2 unusual amino acids, can be synthesized from serine after it has been attached to the tRNA and recognized by specific elongation factor incorporated in the UG stop codon with 6th-actin selenocysteine insertion sequence elements nearby. So, and the pyrrolysine is synthesized from lysine before attached to tRNA and incorporated UAG stop codon with pyrrolysine insertion sequence nearby. So these are the things that you have to have a cognate insertion sequence elements nearby, which can recognize by a specific elongation factor, and the 2 stop codons after that, not UAA, but UAG and the UGA, these 2 nonsense codons make sense right here to be as a sense codon to put pyrrolysine in there. And this is important because you need an insertion sequence element nearby, which can recognize by a specific elongation factor. Just not only UGA stop codon will put that, but you need both of them to put these specific type of amino acids in the protein. And large expenditure of energy helps to ensure the fidelity. and checked before and after formation of a peptide bond. And conformational changes happen when a cognate tRNA is placed in the ribosome. And this release factor recognizes if an incorrect amino acid is incorporated into a peptide, and aberrant peptide is released from the tRNA ribosome, and translation is halted. So that's why this is pretty accurate in this process. So that's pretty much the series of events which the cell use their genetic information to, and this is basically the overall the central dogma of molecular biology, which is replication, transcription, translocation, and this is the one of the most important concept to understand the basic molecular biology, and it will help a lot of different other things and work with each phase and each of these that what happens. So know why and what the gene expression is just as important as knowing the order of steps involved. And don't forget the importance of what and the whys.